Hello, everyone. My name is Rui Castro. I'm an assistant professor at the Faculty of Pharmacy, University of Lisbon, Portugal. It's really a pleasure for me to be here, and I want to start by thanking the Cholangiocarcinoma Foundation and ICRN for inviting me. It's really an honor to moderate this webinar today, which will be on biliary tract cancers. And I'm really excited for our speaker. Her name is Dr. Koshiol. She's from the National Cancer Institute. She's a senior investigator in the Infections and Immunoepidemiology branch of the Division of Cancer Epidemiology and Prevention. And her research, like you see here, it's focused on biliary tract cancers, several really nice aspects on epidemiology, molecular characterization, and she'll also talk to us today a little bit about international collaborations and the importance and the meaning of such for these types of studies. So thank you very much. Jill, please go ahead.
Thank you so much, Rui, for the introduction, and especially to the ICRN and the Cholangiocarcinoma Foundation for this invitation. I'm really excited to have the opportunity to talk with you about some of the work that we've been doing to try to learn about biliary tract cancers. As you know, these are rare cancers, and so they really highlight the need to develop international collaborations to move the field forward and better understand the epidemiology and the biologic processes involved in the development of these cancers, so that we can identify opportunities for prevention and treatment of these cancers. So throughout this presentation, I'm just going to give you some examples of research, and through that process, you'll see how truly collaborative and international this work has to be. And then at the end, I'm going to put out a plea. I'm excited to have this opportunity so that I can make a plea to build a new opportunity that I think might be very important for patients. So just to provide a little bit of background, as you know, gallbladder cancers and biliary tract cancers are rare in much of the world, especially in industrialized countries. But there are hot spots where these cancers really constitute a very important public health burden. And in most of these regions, there's very little understanding as to why the rates are so high. These cancers, because they are rare, particularly in high-income countries, very little work has been done to understand them, which means that we really don't have a good grasp on their associations, even with very classic cancer risk factors such as smoking. And what that means is that there's really a lot that we can do to contribute, and it's important work to better understand these cancers. So today I'm going to tell you a little bit about how we've been working on filling in those gaps in knowledge by describing first the Biliary Tract Cancer Pooling Project, or BitCap, which is a large international pooling project that we've been using to better understand the factors that affect risk of cancer across the entirety of the biliary tract. And then, while I know that this group focuses on cholangiocarcinoma, I hoped you might indulge me while I talk a little bit about gallbladder cancer, a novel association that we've identified with aflatoxin, a study that we've done to better understand the molecular characteristics, and an ongoing cohort study that we have in Chile among women with gallstones. I really want to describe these efforts because I do think that there are important opportunities for collaborations here that I really want to share with the group. And finally, I'll wrap up with a description of some findings on aspirin and biliary tract cancer survival. This is an area that I think really holds great promise, but where we absolutely need to have international collaboration. Starting with BitCap, pooling efforts are really essential to have sufficient power to investigate the association of risk factors with cancer across the entire biliary tract. And that's one of the key strengths of BitCap, which is a pooling project that involves 30 different prospective studies from all over the world, with about 3 million participants and over 5,000 biliary tract cancer cases. Now, this research source is particularly important because there are very few data in the literature, even for classic cancer risk factors and the risk of developing biliary tract cancer. And what data do exist typically are conflicting. So BitCap provides us the opportunity to evaluate the association of these risk factors in cancer across the whole biliary tract. It's really the first effort of its kind, and it highlights how important international collaborations are. This multinational study will provide a much more definitive understanding of the etiology of cancer across the whole biliary tract. So I just want to start with one example, which is smoking. This, again, is a classic cancer risk factor. It's been associated with many different cancers in the body, but very little work had been done to understand how it related to cancers in the biliary tract. Previous studies typically were quite small, usually cross-sectional case control studies. And again, these small previous studies produced inconsistent results with some positive associations and even inverse associations. So to try to better understand what was going on here, my former post-bac fellow, Emma McGee, who's now at Harvard finishing up her PhD, she used data from 26 prospective studies in BitCap with nearly 4,000 incident biliary tract cancer cases and 38.5 million person years of follow-up. And what she found was that, in fact, there did seem to be a different effect of smoking depending on where you looked in the biliary tract. Here you can see the hazard ratios for current versus never smoking at each site within the biliary tract. And just to remind you, hazard ratios above 1 indicate that current smokers have an increased risk of biliary tract cancer. If you were to see a hazard ratio below 1, that would indicate a decreased risk. But what you see here is that, in fact, current smoking was associated with about a 2-fold increased risk of ampulla of Vater cancer and extrahepatic bile duct cancer. The association was a little bit more modest for intrahepatic bile duct cancer, but we really didn't see a whole lot of evidence of association between smoking and gallbladder cancer. That's interesting because shortly after we published our study in BitCap, a meta-analysis came out that reported that current smoking was associated with an increased risk of gallbladder cancer with a pooled estimate of 1.33. Now, this pooled estimate included both case control studies and cohort studies. Case control studies are a great place to start, but sometimes they can be a little bit problematic because of concerns over recall bias or reporting bias. In other words, cases might be more likely to remember or accurately report their smoking behaviors than controls, and that could potentially falsely elevate the estimate. But even in the cohort studies, out of the 10 estimates they provided, 4 of these estimates actually aren't specific for gallbladder cancer. They're for both gallbladder and bile duct cancers combined. And as you just saw on the previous slide, that's a little bit problematic because, in fact, the association of smoking varies across the biliary tract. So if you combine gallbladder and bile duct cancers, then the estimate's going to be skewed according to whichever cancer site predominates in that study. That said, it is worth noting that most of these cohorts are from Japan and Korea. And in Japan and Korea, the risk of gallbladder cancer is higher than in many Western countries. The majority of studies in BitCap are from US and from Europe, which are low-risk populations. So it's possible that in these high-risk populations, there are different etiologic factors. For example, pancreatobiliary maljunction, which is a congenital abnormality that leads to increased risk of biliary tract cancers. That's an abnormality that's more common in Eastern populations, and it's possible that risk factors like smoking might have a different effect in the context of those different risk factors. So to summarize where we're at for smoking, one thing I didn't mention earlier is that when we looked at the amount and duration of smoking in BitCap, we saw the same trends across the biliary tract that we did for current smoking, which I think is important because it suggests that these are real differences that we're seeing across the biliary tract, not just chance findings. And then recent meta-analysis underscores the need for more studies in non-European and North American populations so that we can really try to better understand whether there might be some geographic heterogeneity in the association with smoking. Coffee and tea assumption are also... Oh, yeah, go ahead.
If I can just ask you a quick question.
Sure.
So really nice the studies that you presented. And I was just wondering, because it really struck me to see that there are some differences between the different biliary tract cancers and the effect of smoking. And also This last study that you had presented that did found a higher association of smoking with gallbladder cancer. So I was wondering whether smoking itself, the smoking habits are more or less the same in Japan and South Asia compared to the US and Europe, if you know, because I had the idea that the incidence of smoking habits are heavier in Japan and South Asia, and whether that could also have an effect there. And also if you already have any clue from your studies whether these differences in the different biliary cancers are due to. So what is it different between, for instance, gallbladder cancer and other cancers when we see an odds ratio much higher, for instance, if you have any idea what could be behind that?
Yeah, those are great questions and really good points. You're right that the prevalence of smoking can vary quite a lot. What's interesting about East Asia and South Asia is that those prevalences can also vary a lot by sex, right? So men may be much more likely to smoke than women, but of course you could still have secondary smoke exposure that could affect risk, and that's something that we couldn't address in this study. We just didn't have data from these cohorts to look at that question. It's a really important point that the prevalence of exposure might vary by population, as well as potential on other underlying risk factors. I also think it's a great question to try to understand, and we don't know, is there some biologic difference in how smoking functions in the gallbladder versus other parts of the biliary tract? That's something we don't have an answer to, but I think you're absolutely right. We really need to do some more work to understand what's driving these associations. Is it biological? Is it, prevalence of exposure or other factors that we're not taking into account here?
Yeah, thank you.
No problem. So to give another example of how we've been using BitCap, one thing that we looked at here was also coffee and tea consumption. These are super popular beverages throughout the world, which makes them potentially an important modifiable risk factor for the prevention of biliary tract cancer. But again, their association with biliary tract cancers hasn't been particularly well evaluated. So my former postdoc fellow, Yuhan Huang, used data from 15 prospective studies in BitCap with nearly 900 incident biliary tract cancer cases and 31.4 million person years of follow-up to evaluate the association between coffee and tea and biliary tract cancer risk. And what she found was that for ever coffee drinking, and I have to say, this really surprised us, so we're going to hopefully talk about this in a little more detail in just a minute. We actually saw an increased risk of gallbladder cancer associated with coffee drinking, and we didn't really see much of an association with the other types of biliary tract cancer. For tea, the results were much more like what we expected to see. We saw inverse associations with intrahepatic bile duct cancer and gallbladder cancer. So again, to summarize, we found this really, in our view, strange and unexpected positive association with coffee consumption and gallbladder cancer risk. But we did see a decreased risk of intrahepatic bile duct cancer and gallbladder cancer associated with tea consumption. And really, this gets to the point that you were just asking for smoking. We really need to better understand what's driving these associations. Is there some biologic aspect? Or is it some factor that we haven't accounted for? I didn't go into the details, but we did quite a bit of work to look to see, is there a difference in terms of ever versus never coffee drinkers, in terms of sex, in terms of alcohol, in terms of smoking, since all of these things are often very highly correlated. But no matter how we looked at it, we didn't see this association go away. Except that when we looked in ever coffee drinkers, there wasn't a positive association. So it really was people who drank coffee compared to people who didn't drink coffee. And Ria, I wanted to get your thoughts on that in particular, and especially because when we first started this project, one of the reasons that we thought we would see an inverse association is because coffee drinking is associated with decreased levels of a couple of bile acids, GCDCA and GCA. And previous studies have shown that these bile acid levels are elevated in cholangiocarcinoma patients, compared to controls, and that also that they can promote cholangiocarcinoma cell growth. So we thought, well, if coffee reduces the levels, then we should see an inverse association. That's not what we saw with gallbladder cancer. But there is another study that's come out that found that while in general bile acids are higher in gallbladder cancer patients, GCA specifically was decreased in gallbladder cancer patients. So I don't know if that might provide some evidence of a potentially biological explanation for this. What are your thoughts on that?
Yeah. Well, those are really, really interesting points of view, and I completely agree with you that bile acids should have some effect here. First, let me just tell you that this is really very interesting for me to see this study on coffee and tea and smoking, because these are often kind of disregarded as more general habits on society, but there are millions of people drinking coffee and tea every day, and it's really striking to see that this can have an association, a positive or negative association with cancer, and particularly here biliary tract cancer. So these types of studies I think are really, really important, especially this one that involve a large international collaboration, lots of patients, really good statistics and data. So this is excellent. So going back to the question, I think the bile acids have been shown to associate with cancer, particularly colon cancer and other types of the gastroenterological cancers. And here in particular, so we know that coffee has direct effects on the gallbladder, like on the contraction, on the gallbladder volume. It affects many things, and also the levels of bile acids also change. So I think here, like you were saying, that there are particularly bile acids that have been already associated with increased risk of cancer, like glycosyldeoxycholic acid and others. But you hear so an inverse association that's also really, really strange. But I think that's also possibly the gut-liver axis is also maybe in play here. There's mechanisms of positive and negative feedback going back and forth to the intestine as well. So it might be that changes in bile acid pool and the bile acid species do have something to do with these correlations with developing gallbladder cancer for sure. So really looking forward to know more about this, and hopefully you also elucidate us on some of these findings to see whether it's really something that could be related to this, or maybe the metabolism of cholesterol in bile, or something else.
Yeah, I think maybe a metabolomics study might help a little bit to dig into, is there some biologic explanation involved in this? And hopefully with your knowledge and the knowledge of others in this group, we can really dig in and understand better what we're seeing here. Absolutely. And I agree with you. I think it's super important, just because these are so, everybody drinks coffee or tea. So
exactly.
Yeah. Okay. I wanted to give one more example from BitCap, and this is of a study that we did on reproductive factors. I think that this is important work in particular because the biliary tract, it has this really interesting disparity in the sex ratio, where gallbladder cancers are more common in women than in men, with about a 2-to-1 ratio if you look worldwide, and especially in the Americas. But other biliary tract cancers tend to have a male predominance, which is much more common for cancer in general. That's what you usually see with cancer. So we wanted to try to understand what was driving these sex differences across the biliary tract. So my postdoctoral fellow, Sarah Jackson, looked at this question. We thought that it seemed reasonable that hormones might have an effect, because there have been some studies that have found expression of both estrogen and progesterone receptors in the gallbladder. And there has been a previous study showing that higher levels of estradiol are associated with increased risk of intrahepatic bile duct cancer in women. So Sarah sought to understand how sex hormones might affect biliary tract cancer risk using female reproductive factors as a proxy for sex hormone exposure. And here in the panel on the left, you can see that we found increasing number of live births was associated with an increased risk of gallbladder cancer and kind of a borderline increased risk of intrahepatic bile duct cancer. But we really didn't see any association for extrahepatic bile duct cancer or for ampulla of Vater cancer. In the panel on the right, we found that increasing age menarche was associated with increased risk of gallbladder cancer, intrahepatic bile duct cancer, and extrahepatic bile duct cancer, but only among Asian women, as represented by the light blue circles, not among non-Asian women, represented by the dark blue circles. And there is biologic support for the finding between parity and gallbladder cancer because pregnancy increases the risk of developing gallstones due to both hormonal and physiological changes. The association that we observed with intrahepatic bile duct cancer suggests that estrogens might play an important role in the development of bile duct cancer as well as gallbladder cancer, although that was a little bit of a borderline association, so we need to do some additional work there. Later age at menarche was associated with increased risk of gallbladder cancer, intrahepatic bile duct cancer, and extrahepatic bile duct cancer among Asian but not non-Asian women, which I think is pretty interesting. One thing to note is that Asian women have a lower BMI than women of other ethnicities, and that potentially could delay the onset of puberty. The fact that later age of menarche was only associated with increased risk of biliary tract cancer in Asian women might also suggest a different etiology in Asian women compared to non-Asian women. For example, brown pigment stones rather than cholesterol stones are the predominant gallstone type in some parts of Asia, likely the result of parasitic infections. So again, there potentially could be etiologic differences in this population. And that also fits with the descriptive epidemiology, since that sex disparity that I mentioned is really more predominant in the West, and it tends to be smaller or sometimes even reversed in East Asian countries. We want to follow up on these findings for reproductive factors in a couple of different ways. First, we want to use the data that we have in hand to see how exogenous hormone use is related to biliary tract cancers, so oral contraceptive use and menopausal hormone therapy. But we also want to initiate a new study using biospecimens in BitCap so that we can look directly at circulating sex steroid hormone levels and risk of gallbladder cancer and extrahepatic bile duct cancer in postmenopausal women and men age 50 and above. This is something that we're really excited about because it's the first time we're trying to make use of these existing biologic samples to really go deeper and try to understand, again, what is driving these associations that we're seeing. Some other database studies that we're planning to do include investigating pharmaceutical usage, such as aspirin and NSAID use, as well as statins use. Right now, we're trying to go back to the cohorts to get a little more information so that we can also evaluate diabetic drug use, since diabetes is something that has been associated in different ways with biliary tract cancer. But again, the results aren't always consistent, and we want to try to get a better handle on that. So I thought I'd pause here so that we could discuss, how else can we make use of this resource? One thing I've been thinking a little bit about is how we could engage these cohorts that have DNA or a source of DNA or GWAS data already in hand. Certainly, I know Lewis Roberts has been working for some time on a GWAS of cholangiocarcinoma and gallbladder cancer, but this is a way that we could potentially expand that effort and increase the numbers, which is what you really need for GWAS. So that's one thing I've been thinking about, but do you have other ideas?
Well, yeah, I think definitely starting with GWAS would be great because you have such a large cohort of patients in the study that this will be something great. I know that there's some people that criticize GWAS studies saying that ultimately we'll have like every gene in the genome like associating with something, and that they might not truly reflect the biological effect of the genes that we find in the studies to the disease itself. But I think at some level that's not important, because also if we think on biomarkers and in other stuff, what's important is to find these connections and associations. So definitely this would be something great to start with. And in a related question with this, I also wanted to ask you a little bit more about how the BitCap project and this consortium and database came out to be. I know that you started this a few years ago, and so what type of samples do you have available in the consortium? Do you have the same biological samples at every site of what do you have available, so that we can also like think on what can we do with these tissue samples and think about other possibilities as well?
Yeah, it's a really important question, and it's not entirely straightforward. So each of these cohorts, of course, is its own study, and they have their own data and their own samples. So for example, for the GWAS, what I've been doing over the last 6 or 9 months or so is just to go to the cohorts and say, okay, we're interested in doing this. Can you start by telling us what you have, right? Do you have a source of DNA? There isn't an existing list. Well, I take it back. If you go to the NCI cohort consortium page, there's a way there that you can kind of click on biospecimens and get some sense of what cohorts in the cohort consortium have, but it's a pretty outdated list. So to really get accurate information, you have to go to each cohort and just ask, see if they're interested, and find out if the numbers are sufficient. It's an effort, because it means that you have to get material transfer agreements and ship the samples and do that for all of these cohorts. But I think that it's important, because it's for biliary tract cancers, it's really the only way that we can have sufficient numbers to do things like GWAS, where you have to have big numbers.
Yeah. This could also be a great opportunity for collaborating with the NSCA registry in Europe. So I'm sure you're aware that there's this big also consortium coordinated by Dr. Jesus Vanales from Biodonostia in Spain. It's more on cholangiocarcinoma, but they also have samples and a lot of clinical data in patients on distal cholangiocarcinoma and extrahepatic cholangiocarcinoma. So maybe there's also something there that could add to this project, and then also give more significance and more data for these GWAS studies, or even other studies where different types of biological data are needed. I think you also have imaging data from these patients, right? That's also something.
From these ones, no. So most of the cohorts don't have imaging. They don't have tissues. If they do, some of the cohorts do collect tissues, but they tend to do it for the more common cancers like breast cancer, prostate cancer.
Yeah. Okay.
But I am going to tell you about a cohort just shortly.
Yeah, that's the one I was making. Yeah. Okay. Yeah.
I'll come to it. I'm super excited about it. I think there are a lot of opportunities there. So we'll get to it.
Perfect.
And so kind of moving into some discussion about gallbladder cancer. I bring this up in part just because, BitCap has been great to allow us to evaluate these classic cancer risk factors like smoking, coffee and tea, reproductive factors. But we've also really been making efforts to investigate novel or risk factors that have previously been unsuspected to be involved in biliary tract cancers. And aflatoxin in gallbladder cancer is one example of that. So just to give you a little bit of context, aflatoxins are a family of toxins that are produced by certain fungi found on agricultural crops such as corn and peanuts and certain spices. The main fungi that produce aflatoxin are Aspergillus flavus and Aspergillus parasiticus. And we've actually known for some time that aflatoxins can cause liver cancer, but up until recently, we really didn't expect that it might cause gallbladder cancer. Now, in fact, there are several lines of evidence that support the biologic plausibility of an association between aflatoxin and gallbladder cancer, including experimental data, animal data, and occupational data. In Chile, a previous study identified aflatoxin contamination in food products, such as red chili peppers, whereas at the same time, there's a very low prevalence of strong cofactors for liver cancer, like hepatitis B virus. which we think might explain why there's a pretty low rate of liver cancer in Chile, but high rates of gallbladder cancer in Chile. But even with this evidence, up until recently, nobody had looked directly to evaluate this question in humans. So together with my former postdoc, Leticia Nogueira, and John Grootman from Johns Hopkins University, we tested this hypothesis using samples from a small case control study that we conducted in Chile. And here you can see the actual levels of aflatoxin albumin adducts among population-based controls, gallstone controls, and gallbladder cancer cases. And here in this figure, each bar represents an individual participant, and the red line at the bottom indicates the lower limit of detection of the assay. So bars below the red line indicate we didn't see aflatoxin, and bars above the red line say that we did see aflatoxin. So what you can see is that gallbladder cancer cases had much more detection and much higher levels than either of the control groups. In fact, they were 13 times more likely to have detectable aflatoxin than population-based controls, and 9 times more likely to have detectable levels than gallstone controls. So we thought these findings were super, super exciting, but it's based on one study. And as you can see here, the numbers get very, very small. So we wanted to see, can we replicate this association in a different population? To do that, I used data from the Shanghai biliary tract cancer case control study. And in this study, 32% of gallbladder cancer cases had detectable aflatoxin lysine adducts, compared to only 15% of gallstone controls, for an age and sex adjusted odds ratio of 2.7. So almost 3 times more likely to have detection of aflatoxin in gallbladder cancer cases compared to gallstone controls. Then when we restricted to people who had detection only and looked at the highest quartile of detection compared to the lowest quartile of detection, that odds ratio was 7.6. And in this figure, you can see with the dotted line indicating the gallbladder cancer cases, those levels are shifted up in gallbladder cancer cases compared to controls. Then the next logical step was to compare and see if there was evidence of temporality by looking to see if people who were exposed to aflatoxin were more likely than to go on and later develop gallbladder cancer. To address that question, I turned to the Shanghai cohort study in collaboration with John Menuon from the University of Pittsburgh, and again, worked with John Grootman from Johns Hopkins to test samples from a nested case control study with 84 gallbladder cancer cases and 168 controls selected from the over 18,000 cohort participants. In this study, the odds ratio for detection of aflatoxin was 2, but you can see from the 95% confidence interval that estimate is not very precise. And again, that's because we have a pretty small number of cases that we're working with from this one cohort. Another important point was that the median time to diagnosis was over 18 years, which is a long time. So we wanted to get a sense of whether this estimate changed depending on the time period from baseline blood draw to diagnosis. And we found, in fact, that the odds ratio was pretty similar when we restricted to cases diagnosed below or above that median time from blood draw, suggesting that there actually is a fairly long-term persistent effect of exposure to aflatoxin on gallbladder cancer risk. In addition, individuals who reported having both gallstones and detectable aflatoxin lysine adducts had 9 times the risk of developing gallbladder cancer compared to those without gallstones or detectable or aflatoxin detection. And I think that this is really important because it's the first evidence of a temporal relationship between aflatoxin and gallbladder cancer. So the exposure preceded the development of cancer. And that suggests that aflatoxin abatement efforts really have the potential to help reduce the public health burden of gallbladder cancer in high-risk areas with aflatoxin exposure. So taken together, we saw strong associations between aflatoxin and gallbladder cancer in case control studies, both in China and in Chile. And the data from the cohort study support this association and provide the first evidence of a temporal relationship, suggesting that aflatoxin abatement really might help reduce the public health burden of gallbladder cancer in high-risk populations. And even more importantly, this work is already having an impact. I presented the results of the case control studies to the Chilean Ministry of Health in October of 2016. And that led them to then test Merck's spice blends, which include that red chili pepper previously shown to have potential for contamination. They tested a bunch of spice blends for mycotoxin contamination. And ultimately, they pulled one brand off of the shelves in January 2017 due to high levels of contamination. And I put this up here really kind of as an encouragement, to show that the work that we do is important and that governments like the Chilean government are listening. They want to know what's going on and they can act on these findings in ways that really affect people's health. So the work that we do is important and is having an impact. Ria, I'm going to move on to the next topic. Do you have any questions about this particular section?
No, but I agree with you. This is amazing. This is really a nice example of how what you, as all of us, are doing can really have a big impact on prevention of cancer. And this is really something that I wish would happen also in different areas. So, for instance, I know that aflatoxin is also present in many other products like maize and other products that are stored for a long time. So maybe this can also be something. And what you were talking on the beginning of having some hotspots when gallbladder cancer are also increased, maybe it also relates with environmental toxins like this one and others that you are finding out, and that then we can have a really active role on making them less severe or trying to fund policies and work with the government so that we can prevent some of these more direct and really hard evidences that they contributing for not only blood cancer but others. So and in this case it was you and your team, the scientists who discovered this association, that are also acting as policy recommenders so that these things happen. So do you know about these other food products like groundnuts? I know that groundnuts also can be infected with maize. Do you know if there's this association with other or can foods and that are stored for a long time can also develop mycotoxins and aflatoxin? Do you know if there's any associations that also in different geographical regions might also associate with these contaminations or with these toxins?
Yeah, actually, we don't have evidence yet, but I do think that this might very well be part of what's driving one of the hotspots that's in northern India and in Nepal. So, for example, John Grootman has seen very high levels of aflatoxin contamination in Nepal, and that in the area that's actually right next to the part of northern India that has very high rates. So I strongly suspect that might be happening there. Quite honestly, even in the US, I think there might be areas where this could be a problem. It's interesting because, for example, in Chile, nobody thought aflatoxin was a problem because there are labs that test for aflatoxin all over the place, but they test for exports, food that's being exported out of the country, not food that's being sold in the country. And even in the US, there is a lot of testing done, but it's testing by the federal government. So it's testing food that crosses state lines. Food that is sold within the state is under the regulation of the state itself, and then it's up to the state to do testing or not to do testing. And most states don't really have the funding to do it. So I think in particular in the southern region of the US, and certain populations that are kind of minoritized populations might have an increased risk, in particular because if they are growing their own food or even using kind of traditional medicines. I actually just recently learned that even tree bark can have aflatoxin contamination. And tree bark is actually often part of traditional medicine approaches.
That's true.
I think there's a lot that we don't understand and need to do some more work to even understand what the exposures are in different regions. Yeah.
Absolutely. Yeah, I agree. Thank you.
Okay. So another way to try to gain insight into the potential role of aflatoxin and other factors is by investigating the molecular characteristics of the tumor itself. And to give you an example, NCI partnered with Jesper Andersen's lab at the University of Copenhagen to profile the molecular mutational landscape of gallbladder cancer tumors using whole exome sequencing in 60 tumors from NCI studies, plus 32 publicly available cases with paired tumor and non-tumor data for a total of 92 cases. Then we did some targeted sequencing in about 98 cases. And in a subset, we ran RNA sequencing methylation and somatic copy number alteration assays, just to give us a more comprehensive picture of the molecular characteristics of gallbladder cancer. This figure shows you the mutational landscape of gallbladder cancer tumors based on whole exome sequencing. And the main things that I want you to walk away with are, first, that the overall mutation rate was very low. And second, that TP53 was the most mutated gene, and that's pretty common in lots of different tumors. What's interesting is that even though we saw a lot of TP53 mutations, we actually did not see any mutations in the R249S mutation that shows up in TP53 and is often associated with aflatoxin exposure in hepatocellular carcinoma. So that surprised us. We actually expected to see that mutation and didn't see it, even in people who we knew had been exposed to aflatoxin. One explanation could potentially be that R249S mutation is actually driven by the presence of hepatitis B virus. And in this group, we really didn't have a lot of hepatitis B virus infection. So that might be part of why we didn't see it. And it also leads to a lot of questions about, okay, well, how is it that aflatoxin actually is causing cancer, both in the gallbladder, if it is, as well as the liver? So another way that we looked at this was by looking at single base substitution mutational signatures. And that's just another way to look for evidence of aflatoxin exposure. When we looked at it this way, we found that in fact, about 40% of the cases that we evaluated did have the aflatoxin signature. And these mutations tended to be present in the majority of cancer cells. So when it was there in more than half of the cancer cells, suggesting that aflatoxin really had an effect early in the development of cancer, since it was present in so many of those cancer cells. Another finding was that the ApoBec-mediated mutational signature was more common in tumors that had higher mutational burden. And that's kind of interesting because the ApoBec cytosine deaminases protect from viral infections, leading to questions about, okay, what might be driving the signature that we're seeing in high mutational burden tumors. We then explored molecular subtypes identified through clustering tumors according to genes whose expression was associated with survival. And this was a very small analysis with just 47 tumors. And among these tumors, we found a 95 gene signature that stratified all of the gallbladder cancer patients into 3 different subtypes. 1 that had good survival and 2 that had poor survival. The poor survival subtypes were associated with adverse clinical pathologic features such as advanced stage, immunosuppressive environments such as myeloid-derived suppressor cell accumulation, and also T-cell dysfunction, while the good survival subtype showed the opposite features. So just to give you one example, we used the TIDE immune dysfunction and exclusion algorithm to evaluate predicted immune cell functionality. And one of the things that we found was that the potential for T-cell exclusion was higher in the 2 subtypes that had poor survival compared to the subtype that had good survival. And that's interesting, but this is based on RNA sequencing data, so gene expression. We wanted to dig a little further and see, do we also see evidence of this in the tissue itself, in the actual proteins? So to do that, we looked at digital H&E slide images and in fact found that yes, the subtype 2 that had good survival had higher levels of infiltrates than subtype 3, which had poor survival. Taken together, these findings suggest that gallbladder cancer survival might be dictated more by microenvironment than by something specific, something mutational in the tumor itself. Now, these findings are suggestive, but again, based on just one study and small numbers of cases. So one place that we hope to confirm and expand on these findings is in the Chile Biliary Longitudinal Study, or Chile-BILS. By way of background, Chile has a very high prevalence of gallstones, which is the most important risk factor that we know of for gallbladder cancer. And it also has among the highest rates of gallstones in the world, which makes it a great place to address what I think is really a critical, outstanding question in the field. And that is, among people who have stones, what drives the progression to the development of gallbladder cancer? I think that's an important question because many of the studies that we have show associations with risk factors that are associated not only with gallbladder cancer, but also with gallstones. So take obesity, for example. We know it increases the risk of gallstones. It's also associated with increased risk of gallbladder cancer. We don't know if that increased risk of gallbladder cancer is only because it increases the risk of stones, or whether it contributes something above and beyond the risk of stones. So in the context of stones, when somebody has stones, does obesity still contribute? And that's where we can really address that kind of question here in Chile-BILS. So to address this question, we first identified 29,000 women aged 50 to 74 and did ultrasound screening for about 19,000 to get down to our cohort of 4,726. We're actively following these women through mid-2024. And at that point, we expect to have about 95 to 115 high-grade dysplasia and gallbladder cancer cases. We've really been pretty successful in our efforts with 94% compliance for our year 2 visits. And at this point, 93% compliance with our year 4 visit, which we're actively doing right now. And I just want to pause briefly here to say, this is a reflection of how fantastic the team in Chile is. You don't usually see this kind of really high compliance normally, and they've managed to achieve it in the midst of a global pandemic. I cannot say enough about how creative and how dedicated our team is to be able to get this kind of response. And also for the participants who are willing and able to be part of this study. It's really pretty phenomenal. And during the whole course of the study, so far we've had more than 2,000 people go to cholecystectomy to have surgical removal of their gallbladder. To give you a sense of the baseline characteristics, 55% are age 50 to 59, 53% have 8 years or less of education. About a quarter of the population identifies as being Mapuche, and that's the predominant ethnic group in this region, which has previously been shown to have extraordinarily high rates of gallbladder cancer. 60% of the cohort is obese, 25% has diabetes, and 6% cardiovascular disease. You can see kind of the skew of this group of individuals with prevalent gallstone disease. And then an important point is that about a third of the cohort had previously experienced biliary colic at baseline. That's important because it means that 2/3 of the cohort hadn't had colic, so really was pretty asymptomatic. And that is good because we wanted to get a good representation of the entirety of gallstone disease. And in fact, most people who have gallstones don't know that they have them. And so we achieved that in this cohort, which is really important. We also have extensive epidemiologic data, biospecimens, and digital images in Chile-BILS. Just to give a few examples, we have baseline questionnaire data on 99%, baseline blood draw data on 96%. We have an expanding repository of digital ultrasound images and H&E slide images. Those ultrasound images are from baseline year 2 and year 4, so we have serial images, and a growing number of surgical specimens. In addition to what I'm showing you here, we have FFPE tissue for over 90% of those who've gone to have cholecystectomy. So this is really a pretty important resource that I think is going to provide a lot of information about the natural history of disease. And even though we are continuing with the study, we haven't finished it yet, field work is ongoing, we've wanted to start making use of this massive amount of data and biospecimens that we've collected. So one way that we did that was to try to understand, why are the rates of gallbladder cancer so high in people with Mapuche ancestry, as I mentioned before. So we know that Mapuche ancestry is associated with gallbladder cancer. Previously, we've seen that circulating inflammation markers are associated with gallbladder cancer. And so we wondered, could this association with gallbladder cancer in Mapuche individuals be driven in part by differences in inflammation. So we sought to see if inflammation is greater in Mapuche women than in non-Mapuche women. To address this question, my postdoc Sarah Jackson measured 92 circulating inflammation markers in a subset of people from the cohort, 200 women who identified as Mapuche and 200 non-Mapuche. And she used a statistical method called sufficient dimensional reduction to understand whether linear combinations of these markers could identify which women were Mapuche and which were not Mapuche. In this figure, the red dots indicate the non-Mapuche women and the blue squares indicate the Mapuche women. So as you can see, these linear combinations of all of these inflammation markers really did a pretty good job of discriminating between Mapuche and non-Mapuche women. What that's telling us is that the inflammatory profile is different between Mapuche and non-Mapuche women. However, the way that we interpret these differences is really important and is actually kind of a point of contention. For example, an article was recently published that criticized our work because they felt that our interpretation reinforced social constructs of race and ethnicity as if they had a biologic basis, when in reality, these differences could be due to a lot of different things. And in fact, that criticism is not without merit. Sometimes we scientists have a tendency to jump to genetics as an explanation for differences in racial ethnic groups, but these differences could instead be due to lack of access to care, behaviors, diet, or geographic factors, such as whether somebody lives in an urban or a rural area that could differ according to ethnicity, or even stress due to the social consequences of race ethnicity that potentially could have a biologic impact, such as increased inflammation. And so for these reasons, I added some questions about perceived discrimination to the year 4 questionnaire in the cohort. It's not perfect. There's a lot of different things that we could do. And because we're in the middle of the year 4 visit right now, we don't have enough data to look at this yet. But my hope is that this kind of information will begin to give us a little bit better sense of what's driving this disparity that we see between Mapuche and non-Mapuche individuals. Some of the longer term questions that we hope to address include things like, what factors predict the risk of dysplasia and gallbladder cancer? And coming back to what I mentioned at the beginning of the section, for example, obesity. Do we see that association with obesity among these women who have gallstones? We also want to dig more into this question of aflatoxin. For example, do we see aflatoxin associated with gallbladder dysplasia, high-grade dysplasia and low-grade dysplasia, earlier stages of disease, as well as cancer? And how strongly are circulating inflammation markers associated with gallbladder dysplasia and cancer? And could they be used to help us figure out who to prioritize for surgery? Who's going to benefit the most among people who are waiting to have surgery? So ChileBills is a particularly valuable resource because it's really the first well-designed epidemiologic gallstone cohort in a region with high risk of gallbladder cancer. There's a high prevalence of understudied exposures. As you've seen, we have really extensive biologic specimen collection that's going to help us better understand the natural history of disease and how different exposures are related at each stage of disease, and to conduct molecular efforts to better define what that process looks like. We also have extensive imaging data with ultrasound images over time and digital slide images. And there's great potential for translation of these findings to public health policy. That potential is important not only for Chile, where people have to wait sometimes for years for cholecystectomy, but I also just recently heard that this is a problem even in the UK, where the waiting list is well over a year at this point and expected to be even out to 5 or 6 years over the course of the next few years. So this is really an important issue of who to prioritize for surgery in places where the waiting lists are long. That's not unique to Chile, but is important in other parts of the world as well. So I wanted to share this with ICRN because I think this is a really rich data source. There are many opportunities for collaboration here, many opportunities to better understand this really very highly neglected cancer in the biliary tract. And I wanted to take this chance to get some feedback as to whether, can this resource also help inform us about bile duct cancer? We have all of this tissue, all of these images, And in fact, gallstones are a risk factor for bile duct cancers as well as gallbladder cancer. So, Ree, what do you think? Are there opportunities here?
Absolutely, absolutely. First off, this is an amazing study, really lots of data, lots of patients, lots of information about their clinical history, extensive collection of samples, like you were saying. So this is really something that we should take advantage of to find out as much as possible to benefit these patients and also other patients, like you were saying, for other types of biliary cancer. I think this is a highly valuable resource, for instance, for doing omics analysis, metagenomics, proteomics, and different types of these high-end recent analysis to find out really in detail, to pinpoint specific things that might associate with, like you were saying, the probability of someone developing a different or a particular type of cancer quicker or slowly throughout time for prioritization for surgery, like you were saying. And there's definitely here a lot of space for collaboration, and I think this is why these kinds of initiatives like this one from ICRN and the Cholangiocarcinoma Foundation is great, so that we can bring this to new audiences that can also benefit from studying these patients and these samples. I also wanted to ask you here, related to this, if you are also planning, or whether these patients are also going through surgery and you are also following them up after surgery or after chemotherapy, so that you also gain information on whether certain treatments are more have more efficacy than others, and things like that.
Yeah, so the answer is yes and no. For everybody in the cohort, we are linking to the death registry, so we do find out when people die, and for gallbladder cancer cases, we are going back to the medical record to try to get information about what treatment was used. For people, but the majority of people who go and have cholecystectomy, they don't have cancer, they don't have dysplasia, they might have intestinal dysplasia or something else. And those individuals who are not following, I actually do think that there are opportunities there that could be valuable. For example, with these inflammation markers, we do see changes in inflammation markers prior to the development of cancer, but we really don't know what happens after you remove the gallbladder. There have been a few small studies that have said that things often go back to normal, but not always. And in fact, Caterina Ferrecchio, who is the primary PI in Chile on this study, she kind of hypothesizes that there might be systemic changes in inflammatory profiles due to this prolonged chronic inflammation in the gallbladder that might not be resolved even after you have that gallbladder removed. And we don't know the answer to that question. So it's something that I actually would really like to do, but have yet to be able to do in this group.
Perfect. Yeah. Thank you.
All right, well, let me move on to the very last part of the talk, which I think this is a super important part of the talk. So everything I've shown you so far has required substantial investment involvement from collaborators from all over the world. And I want to end this presentation with a plea for a new international effort to really try to establish whether aspirin can improve biliary tract cancer survival. By way of background, I became interested in aspirin and biliary tract cancer survival, in part because of a study conducted by Ziwei Liu, who's a tenure-track investigator in our branch. He found that 8 circulating inflammation markers were associated with overall gallbladder cancer survival, and 5 of those 8 markers were actually associated with early stage 1, stage 2 gallbladder cancers, as well as with advanced cancers. 7 out of the 8 markers had higher levels associated with poor survival. One of the markers, trail, actually had higher levels associated with improved survival. And the differences in the first quartile versus the 4th quartile of these markers varied from about 8 to 10 months. So these aren't trivial differences in survival that we're seeing here, according to these inflammation markers. the fact that so many of these markers were really strongly associated with survival made me wonder if anti-inflammatory drugs might help improve survival. So to address this question, we turn to the United Kingdom Clinical Practice Research data link, and found, in fact, that there does seem to be a beneficial effect on survival with post-diagnosis aspirin use, and that's regardless of where you look in the biliary tract. So here you can see in these little figures, the gray line indicates survival in individuals who used aspirin post-diagnosis. The black line indicates survival in people who did not use aspirin. What you see, for example, with gallbladder cancer is that at 12 months, so at 1 year, this post-diagnosis aspirin users had 59% survival versus only 27% in non-users. And that pattern was very similar at every anatomic site in the gallbladder. We then followed up on these findings using registry-based data in Taiwan, and again found very similar findings with dramatic improvements in survival for all types of biliary tract cancer with the use of aspirin after cancer diagnosis. I think these findings are really important because they suggest that aspirin might be a cheap and readily available way to dramatically improve the survival in biliary tract cancer cases. But it is important to be cautious because we can't rule out the potential that these findings might be biased by things like healthy user effect or healthy adherer effect. In other words, that more health conscious individuals might be more likely to take aspirin or might be more likely to adhere to prescribed treatments. We did try to account for these kinds of biases by adjusting for things like high exercise or healthy diet as an indication of healthy users, and participation in cancer screening and influenza vaccination as an indication of healthy adherers, as well as functional status measured by the presence of pneumonia or dementia or comorbidities, including organ failure. None of these things really changed the magnitude of the effect that we saw, which is reassuring. It suggests that these findings aren't just due to selection bias, but there's always that potential for residual confounding. So to really establish whether aspirin can improve biliary tract cancer survival, I think we need a clinical trial. And that is exactly the kind of international collaboration that I hope ICRN and INSCA might be interested in. And this is what I'd love to just pause here and finish out this discussion with, can we make this happen? Is it feasible?
Yeah, I agree. I think this would be great. I agree with your opinion, and this is something that we really deserve to know more about. There has been a lot of observational findings, like you said, suggesting that indeed aspirin could be efficient in preventing biliary tract cancers, and especially gallbladder cancer. But we really need a really good controlled randomized clinical trial. There are, I think, some clinical trials ongoing with aspirin for other cancer types, but not for biliary tract cancer. So this is a great opportunity for such a study. And also, there are some also things that I think we should have into consideration. For instance, age. There has been some reports that people at an older age do not appear not to benefit as much from aspirin as younger people in terms of cancer prevention. And there's a lot of, so this would be something that we could also address in such a clinical trial. I think there was a recent trial suggesting that for HER2 negative breast cancer, aspirin didn't appear to have any protective effect. But in contrast, like you were saying, there are so many observational positive findings suggesting such an effect that we really would need a clinical trial to do this. And like you were saying, this would be the prime example of what international collaborations and bringing forces together to do this at a large scale. This would really benefit, and because this would be something really important for not having these many patients, or for preventing disease in a lot of people. And I think this, like you were saying, is something that ICRN, the Cholangiocarcinoma Foundation, and also the European Association working on cholangiocarcinoma and biliary tract cancers should join forces to really put together. That would be great for sure.
Yeah, I also wondered if there's something that's kind of in the works, if there's a clinical trial that people are working on designing now, if we might build in an aspirin component.
That will be another hypothesis. I am not aware of any clinical trial being set up, but that's something that we should definitely try to find out more, because I think this would be really, really important, like you're saying.
Yeah. I remember from the last ICRN meeting, at the annual meeting, there was a discussion about the challenges of trying to do international patient recruitment. But I do think that this is a place where we need that kind of work, if it's all possible to make it happen.
Yeah.
And these kinds of findings, you have to know.
Exactly. And there's a lot of people like yourself and others in Europe, like the people working at ENSCA, that already have experience in these types of studies and in recruiting patients. So that somewhat also ameliorates that effect of recruiting patients, because we'll already have experience in that, and could be also a positive point there. I think we are running out of time, but this was an excellent webinar. I really need to thank again the Cholangiocarcinoma Foundation and ICRN, and also you, Jill. This was amazing for me. I have to confess, I wasn't that in your known of your work and really in depth, and this was just amazing to see it. Really, how the amount of studies and the different things that you are doing and that are having a real impact in the lives of these patients or people that could be affected by this disease, like we saw with your work together with health authorities, and this was an excellent webinar, and I thank you also for that.
Well, thank you so much, Rui, and again for ICRN and the Cholangiocarcinoma Foundation. Let me just very, very quickly in like 10 seconds.
Yes, please.
You know, I have so many people that none of this stuff is done in isolation. All of it really takes a huge team of collaborators from all over the world. I do want to specifically highlight the fellows and the former fellows who were instrumental to all of the studies I showed today. And then just lastly, you know, to thank all of you for your time and your attention and your interest in this really important topic. It's only by working together that we can move the field forward. So thank you, everybody.
Thank you.